Bruno Lomonte from Costa Rica. Bruno is renowned not just for his fantastic work on a variety of snake venoms, applying venomics and antivenomics to try and understand the venoms, develop new antivenoms, but he's also renowned as perhaps the nicest guy in the world of toxinology. And it's a really great pleasure to have him here. So he's going to tell us about some of his work on coral snake venoms. So take it away, Bruno.
Thank you so much. Hello, everyone. Good morning or good afternoon. Thanks for the invitation and the opportunity to present this work on Micrurus or Micrurus diastema venomics, unveiling the dichotomic arsenals of New World coral snake venoms, which I'm presenting on behalf of a group of researchers from Mexico and Costa Rica. So the ancestors of elapids invaded the New World, crossing the Bering Land Bridge some 22 million years ago, and evolved an impressive radiation or speciation with over 80 different species in the genus Micrurus, one species in Microroides, while Leptomicrurus is still in debate. So this was an impressive radiation, and traditionally, the venoms of the coral snakes in the Americas have been difficult to study and to analyze because these snakes have poor survival in captivity, and they provide very, very low amounts of venom during extractions. However, with the omics technologies have provided new opportunities to study these hard to get venoms, and especially proteomics, which we have used in our laboratory to focus in different types of venoms, including those of coral snakes. So of the about 80 species of Micrurus snakes that we have in the Americas, about 20 of them have had their venoms analyzed by proteomics strategies. So that represents about a quarter of all the species. So we must bear in mind that still there is a lot to discover and to know about these venoms in this group of snakes. First, in studying the coral snakes of Costa Rica, it was found an interesting dichotomy of venom profiles, in which, in Costa Rica, it's a small country in Central America, there are 6 recognized species of Micrurus. We have 3 of them inhabit the Pacific region of the country, over here, and other 3 in the Caribbean region. Both regions are separated by a central range of mountains. And so when the venoms of these snakes was studied, we found an interesting observation in which some of them have a profile, a composition in which 3-finger toxins are predominant, are highly abundant. Here represented with this orange slice of the compositional pie. You can see these 3 species, they are predominant in this orange slice, while the others have less 3-finger toxins, but more phospholipases A2. This is the purple slice. Interestingly, the species can inhabit in the same region of Costa Rica and still have these divergent phenotypes. And these observations could be also extrapolated to, to other coral snakes from other countries. For example, from Colombia, I can show you here Micrurus bipartitus, which is predominant in 3-finger toxins, whereas Micrurus dumerili, which has more or less the same geographical distribution, has a totally opposite balance between these two types of components. So this is a PLA2 rich venom, and so on. So some time ago, we proposed that probably during the evolution of this group of coral snakes, there was a divergence in the expression of proteins in the venom, in which some of the species came to produce more PLA2s and others came to produce more 3-finger toxins. So these 2 phenotypes have some interesting correlation with the geographical distribution of the species of the snakes, in which coral snakes that inhabit more in the north of the American continent, they have this PLA2 predominant phenotype, whereas in the south, it's more three-finger toxin rich. And there is a mixture in the center. In the central region of the continent, we have both types of phenotypes, or I mean, species that express both types of venom phenotypes. So on this occasion, I would like to share our data from another Micrurus that we were able to study, Micrurus diastema, which is a coral snake distributed in the southern regions of Mexico, down to the northern countries of Central America, including Guatemala, Belize, and Honduras. So the venom was analyzed by two proteomic profiling strategies. We used, on one hand, the Venomics protocol that was developed by Calvete's group in Valencia, Spain. And also we used the shotgun approach with a direct digestion of the whole venom without any decomplexation. The chromatographic profile of this venom immediately reflected that this was going to be one of those PLA2 rich venoms by judging, by comparing with other coral snakes venom we have studied. And actually, it's a relatively complex, the number of peaks. And actually, it was confirmed by the proteomic data that this species produces a PLA2 rich venom. Well, see, this is the compositional pie. And as you can see, in the purple, you have PLA2s, which is about 60% proteins in the venom. There is, of course, a considerable amount of three-finger toxins too, but this is the balance that we obtained. There are some other protein families with relatively lower relative proportions of the total composition. So it is interesting to add more data to these findings that we have been collecting and other groups on the Micrurus snakes. For example, if we position the two phenotypes on the phylogeny, or the partially known phylogeny of this group, which is kind of complex, we can see that three-finger toxin-rich species are more basal in the phylogenetic tree of these snakes, whereas those with the PLA2 predominant phenotype tend to be in more derived branches of the phylogenetic tree. So actually, on this basis, we had proposed that probably the ancestral state was three-finger toxin predominant phenotype. And actually, this was recently confirmed, or it was in agreement, when the venom of Micruroides eurixanthus is only one species for this genus, which is basal to the whole tree. When this was studied by Dr. Melissa Bernard and her colleagues in Mexico, they confirmed the predominance of three-finger toxins in this venom. So in the case of Micrurus diastema, which we're talking about here, it was confirmed also that it is a PLA2 rich venom, and it's positioned in the more derived branches of this phylogenetic tree. So again, following this kind of rule or trend in the findings. But still, it's important to remember that there are many, about 60 species of Micrurus that need to be investigated to get a more complete picture. One interesting toxin in this group of snakes was discovered in the venom of Micrurus tener, which is the Texas coral snake. This toxin is a heterodimeric complex and is capable of binding to the acid-sensing ion channel 1 and induce pain. So this toxin we have been able to find in at least three other species of Micrurus, which all of them are this PLA2 rich phenotype. Here I'm representing with this purple and orange, the two phenotypes. And on the contrary, all the venoms that are rich in three-finger toxins, this protein has not been found. So it seems that it's correlated or associated with this shift in phenotypes during evolution, perhaps. And in the case of Micrurus diastema, so it follows the same rule. So the venom is PLA2 rich, and this pain-inducing heterodimeric complex was also detected in this species from Mexico and the north of Central America. So what are the implications of this venom dichotomy for serotherapy? Some time ago, it was shown previous studies that the antivenoms produced both in Mexico and in Costa Rica are capable of neutralizing the coral snake venoms, the lethality in mice from species that belong to the same group. So the horses are immunized with a PLA2 predominant venom, Micrurus nigrocintus. And then the antivenoms, both of them can neutralize well species that belong to the same group of this dichotomy, but they do not neutralize, or maybe just poorly, venoms that belong to the other group. So this is the main implication. And in the case of Micrurus diastema, so it was confirmed that it followed the same rule. Both the Mexican antivenom was neutralizing the lethality, and the same happened with the Costa Rican antivenom with Micrurus diastema. So I would like to summarize some of these trends or emerging rules that we're observing with the venoms of the Micrurus by applying this proteomic analysis and biochemical and toxinological studies, which are the following. So in general, these venoms have very few protein families, and always three-finger toxins and PLA2s are the vast majority. But they have a different balance. Some of the species, there are three-finger toxin skewed predominant or PLA2 predominant phenotypes. These phenotypes relate with the partially known phylogeny of this group of snakes, and it seemed that the three-finger toxin rich was the ancestral state, which was followed in evolution by diversification of PLA2s and increase in the amounts, in the concentrations, and in relative proportions in the venom. There is this north-south polarization in the two phenotypes, but still there are many venoms pending to be studied. See if this trend holds true after more species have been studied. And in the middle of the continent, we have a coexisting phenotypes, a mixture of the two. Then in general, the PLA2 rich venoms are usually more complex. They have more protein peaks, more proteoforms in average, as compared to those producing more three-finger toxins. And finally, this dichotomy between these two types of toxins has a relationship with the immunological divergence, and this has its obvious implications in the for antivenoms and serum therapy of envenomings produced by these snakes, which are not very common. They're not it's not very frequent to see people bitten by coral snakes, but still those envenomings can be very serious and potentially lethal. And as I mentioned in the beginning, so it was a pleasure to perform this study in collaboration between two institutes, Instituto de Biotecnología, or Biotechnology Institute from UNAM, Cuernavaca in Mexico, and Instituto Clodomiro Picado at the University of Costa Rica by Dr. Melissa Bernard, student Manuel Yañez, Dr. Edgar Neri, and Professor Alejandro Lagon. And on this side, student Marco Ruiz, Dr. Julian Fernandez, and myself. And I thank you very much for your attention.
Questions from the floor? Juan, do you have a question?
Hope the time was okay.
Hi, Bruno.
Hey.
Glad to see you, and congratulations. Nice talks like ever. I have two short questions. One, remind us what's the last ancestor in Asia, the last common ancestor to the micrurus in Asia.
Yeah.
And if you know, and if the venom is known, if it is also a three-finger toxin rich, or could be that the trend changed with passing the Bering Strait.
Yes, that's a very good point. So in the old world, there are coral snakes. They have the genus Cynomicrurus or Cynomicrurus and Calliophis. I believe there is only one proteomic study with Calliophis. Now it escapes my memory. I think it was more PLA2 biased. I'm not so sure, but yes, I think it was like that because it was kind of surprising because I would expect more of them. Yes, cytolysins, three-finger toxins, which we don't have in the American continent. This is also very interesting, right? No one has found cardiotoxins in micrurus, and yeah, but then probably I don't know. Many things could have happened with from the ancestor to the modern present day. This Calliophis is only Calliophis virgatus. I think is only one has been studied in this regard.
Mm-hmm. Mm-hmm. And the other was, if I remember correctly, the three-finger toxin rich and the PLA2 rich phenotype cannot be distinguished from the lethal activity, at least that they have. So they have similar LD50s. So
right.
Anything that could be perhaps be diet selecting these phenotypes. They have different diets, or is I think it's difficult because not so much is known about the diet, but I have not followed lastly this field, so perhaps you have a better answer.
No, I don't have much further information. It is difficult to try to rationalize what does this dichotomy means, right? Because yeah, the diets of coral snakes, as far as it is known, many of them are ophiophagous. They eat small snakes, and some of them live in the mud and water, and they eat some. For example, here in Costa Rica, they eat Cymbrancus, which is a small eel fish, and there are not many studies to determine the toxicity, the lethality, or the paralyzing activity of the venom on the natural prey. So we rely on studies on mice, as you well know, that they do not represent nature, right? These snakes do not feed on rodents, as far as we know. So it's difficult to understand. So when actually, yeah, with your help, we were looking at the lethal potencies of the venoms in mice, and there was no clear understanding of what this dichotomy means. So there was comparable average toxicities in the two groups. So
okay, we shall continue the discussion in Abu Dhabi.
Okay.
One last question from Jan.
Jan Titgat here. Thanks, Bruno, for the presentation. The north-south polarization you mention and observe, does it also stands if you only consider, let's say, the three-finger toxins of all the micruruses, or if you then compare the sequences, is such a polarization then also present, and just alike for the phospholipases, if you only consider that particular set of peptides or toxins?
Okay, you mean like if you compare the sequences in the north and the south of the same group of proteins?
Yes.
Oh, it's a good point. We have not tried to do that, but would be very interesting. Yeah, we have not.
Thank you, Bruno. I think in the interest of time, we need to move on. So could you join me in thanking Bruno for a great talk?